Overview of embryology. Embryology is the branch of biology that deals with the development of an organism, from the fertilization of the egg to the fetus's formation and beyond. It is an essential field of study in understanding how life begins, how organisms develop, and how abnormalities can arise during development. The science of embryology touches many disciplines, including genetics, molecular biology, developmental biology, and medicine. 1. Definition of embryology. Embryology is primarily concerned with studying the developmental stages of an organism before birth. In humans, embryology focuses on how a fertilized egg, zygote, develops into a fully formed individual through a series of complex processes like cell division, differentiation, and morphogenesis. 2. The importance of embryology. Embryology is essential for several reasons. Medical applications. It is vital for understanding developmental defects, birth defects, and how diseases manifest during pregnancy. Assisted reproductive technologies, the first ART. Embryology is crucial for the development of IVF, in vitro fertilization, egg freezing, and embryo transfer techniques. Genetics and gene expression. Understanding how genes are regulated during development and how mutations can cause diseases is a major application of embryology. Stem cell therapy and regenerative medicine. Research in embryology leads to advancements in stem cell therapy, which has potential applications in treating degenerative diseases. Third stages of human development in embryology. Human embryology describes the developmental process from conception to birth. The development is traditionally divided into the following stages. Fertilization, the process where a sperm and egg combine to form a zygote. This single-cell organism contains all the genetic material necessary for the development of a human being. Cleavage, the first few rounds of rapid cell division that occur after fertilization without an increase in the size of the embryo. The early divisions result in a cluster of cells known as the morula, followed by the formation of the blastocyst. Gastrulation, a critical stage where the simple bilayered embryo becomes trilayered, forming the three germ layers, ectoderm, mesoderm, and endoderm, which will later develop into different body structures. Organogenesis, the process where the three germ layers differentiate and develop into various organs and tissues of the body, such as the brain, heart, kidneys, lungs, and limbs. Fetal development. Following the formation of the organs, the fetus grows and matures until it is ready for birth. During this period, organs and structures further specialize to perform their specific functions. Fourth, historical development of embryology. Embryology has evolved significantly over the centuries. The history can be divided into several key periods. Ancient beginnings, pre-1600s. Early philosophers like Aristotle were some of the first to propose ideas about embryonic development. In his work, Generation of Animals, Aristotle theorized that the fetus developed from menstrual blood and proposed that the development of organisms followed a process of epigenesis. Galen, 129 to 216 AD, a prominent Greek physician, believed that the fetus was nourished by the mother's blood and that the heart was the first organ to form in the embryo. 16th to 18th century, early scientific observations. William Harvey, 1578 to 1657. Harvey was one of the first to propose that the embryo does not simply grow into its form from a preformed miniature adult, the idea of preformation. Harvey's observations on the development of chicks and other animals helped him conclude that embryos develop from an egg. Marcello Malpighi, 1628 to 1694, an Italian biologist who made significant contributions by using a microscope to observe the development of the chick embryo and other animals. He was one of the first to describe the early stages of chick development. 19th century, the birth of modern embryology. Carl Ernst von Baer, 1792 to 1876. A pivotal figure in the foundation of modern embryology. Von Baer demonstrated that all embryos of different species appear very similar at early stages, and he formulated the concept of the three germ layers: ectoderm, mesoderm, and endoderm. This discovery laid the groundwork for understanding how complex organs and tissues form during development. Antony van Leeuwenhoek, 1677. He made the first detailed observation of sperm using a microscope. His work provided insight into fertilization. Charles Darwin, 1809 to 1882. Darwin's theory of evolution also has significant implications for embryology. The idea that development could help explain evolutionary relationships between species has been an area of active research since the 19th century. 20th century and beyond technological advancements, genetics and molecular embryology. In the early 20th century, genetics became integrated into embryology, with Gregor Mendel's principles of inheritance providing a foundation. Later, the discovery of DNA and the genetic code revolutionized how scientists understood the genetic basis of development. In vitro fertilization, IVF, the first successful IVF procedure, which led to the birth of Louise Brown in 1978, marked a significant milestone in embryology. IVF allows scientists and doctors to study the early stages of embryonic development outside the human body. Stem cell research. In the late 20th century, the discovery of embryonic stem cells opened the door to potential regenerative therapies. Stem cells are unique in that they can differentiate into any type of cell, which has led to innovative treatments for various diseases. Fifth, key concepts in embryology. Gametogenesis. This refers to the process by which gametes, sperm and eggs, are formed. In males, the process is called spermatogenesis. In females, it is oogenesis. Both processes involve meiosis, a type of cell division that reduces the chromosome number by half. Fertilization. The union of a sperm and egg results in the formation of a zygote, which marks the beginning of development. The zygote has a complete set of chromosomes, half from the mother and half from the father, and carries the genetic information for the embryo's development. Differentiation. Differentiation is the process through which cells become specialized to perform specific functions. After fertilization, the initial cells of the embryo, called blastomeres, begin to differentiate into various types of cells, which will form tissues, organs, and systems. Morphogenesis. This term refers to the process by which the shape of tissues, organs, and the overall body plan are formed. It involves cell movement, division, and differentiation, and it is driven by complex signaling pathways. Apoptosis. This is the process of programmed cell death that plays an essential role in shaping organs and tissues during embryonic development. For example, the fingers of a developing hand are formed by cells dying in a process called interdigital apoptosis. 6. Modern embryology, technologies, and research. In recent years, advances in technology and research techniques have led to the rapid development of embryology as a discipline. Some of the most important advances include microscopy and imaging. The use of time-lapse microscopy allows embryologists to observe and record the development of embryos in real time. Techniques such as confocal microscopy and fluorescence microscopy allow for detailed imaging at the cellular level. Pre-implantation genetic diagnosis, PGD. PGD is used in conjunction with IVF to test embryos for genetic disorders before implantation. This helps in identifying embryos free from specific genetic conditions, enhancing the success of fertility treatments. Gene editing and CRISPR. The advent of gene editing tools like CRISPR/Cas9 allows scientists to modify genes within embryos. This has profound implications for understanding gene function and potential therapies for genetic diseases. Stem cell therapy. Embryonic stem cells have the potential to become any cell type, which makes them invaluable for studying disease mechanisms and creating treatments for conditions such as Parkinson's disease, diabetes, and heart disease. 7. Ethical considerations in embryology. Embryology also raises important ethical questions. Some of the major issues include stem cell research. The use of embryonic stem cells for research raises concerns about the destruction of human embryos. Cloning. Reproductive cloning and therapeutic cloning are subjects of debate, particularly concerning the ethics of creating embryos for scientific or medical purposes. Genetic engineering. Gene editing, particularly in human embryos, has sparked significant ethical discussions about the potential for designer babies and unintended consequences. 8. Future of embryology. As technology continues to evolve, the future of embryology holds vast potential for improving human health and understanding biological processes. Organoids. Miniature organs derived from stem cells, genome editing, and advanced fertility treatments are just a few of the exciting developments on the horizon. Conclusion. Embryology is a foundational field in biology, medicine, and reproductive science. It provides crucial insights into how life begins, develops, and diversifies. Modern advancements have allowed researchers to manipulate and study embryos in ways never before possible, leading to new medical treatments and a deeper understanding of human biology.